This video is going to discuss the first major stage of gene expression, transcription, where we build a mRNA molecule based on the sequences of bases in a DNA molecule. So to build our mRNA, we need a really important enzyme, RNA polymerase. So it's the job of this RNA polymerase to actually build our primary mRNA transcript, that initial mRNA sequence, which is a faithful copy of all the bases within the DNA. So what RNA polymerase does is it comes along, unwinds the double helix, breaking apart those hydrogen bonds that keep the bases together. So it has the DNA to act as a template. So RNA polymerase can then read the DNA sequence and use the base pairing rules to build up our primary mRNA transcript. So where it sees a C in the DNA, it has a G, a C, a T, an A, and because T in RNA is replaced by uracil, if there is an A in the DNA, there will be a U in the mRNA. And at the end, we have our primary mRNA transcript. But our mRNA is not quite ready to leave the nucleus just yet. We have to do a little bit of processing before we can use our mRNA as your basis for translation for protein synthesis. So before it leaves the nucleus, it first has to undergo splicing. So splicing is going to remove anything that interrupts or is not needed. Remember, DNA contains a mixture of coding and non-coding regions. So anything that's not relevant to building the protein has to be removed from your primary mRNA transcript. That which is a faithful copy of the DNA molecules would contain both the exons, the coding regions, and those introns, the non-coding regions that need to be removed. So introns are removed, exons are joined or spliced together, and that is how we make our mature transcript, the one that we can use to do translation. So when we do this, the order of the exons are completely unchanged, so we still have the information that was contained within the DNA molecule of which order to add the amino acids to build our protein. But because the sequences are unchanged, it doesn't mean we can't still build many different types of proteins from the same information. So there is a process known as alternative splicing, where we can join together different exons, which would result in the translation or the building of an alternative protein. So we can see here we've built our primary transcript, we've removed the introns, and we have the different exons, the different coding regions that need to be expressed. So we can join them all together to make protein A. Maybe we can skip exon 3 and make protein B, or alternatively skip exon 4 and build protein C. So from the same gene, the same DNA sequence, we can actually build different proteins. Okay, so that's everything you need to know about the transcription process. Happens in the nucleus using RNA polymerase to build the mRNA molecule. So let's try some past paper questions. So the first one, you've got a diagram that shows the stages in the synthesis of a polypeptide, the basis of a protein, and you're asked to name the enzyme that catalyses stage 1 of this process. And question 2, you're asked to explain why the mature mRNA transcript was shorter than the primary mRNA transcript. Pause the video, come up with your answers, restart to see if you were correct. Well, starting with part A, question 1, The enzyme, so stage 1, as we can see from the diagram, is going from your DNA to your primary mRNA transcript. So that must be your RNA polymerase. You remember to be specific and add RNA. The polymerase enzymes, they build, so you have to be specific about what they actually build. So remember, we came across DNA polymerase during DNA replication. And question 2, to explain why the mature mRNA transcript will be shorter than the primary. Well, that's because the primary transcript has to undergo splicing, so whole regions, those introns, are going to be removed, which means you've got a shorter molecule. Or the mature mRNA transcript only contains exons, whichever way round you want to express that. So again, I hope this has helped break down the topic, and translation will be up next.